So today we'll talk about curating biomedical molecular data at a scale. In the last UGM, we talked about like preliminary ways to curate biomedical molecular data, but since then we have come a long way to do it at scale, and that's really important, as you must have seen by now from Chef's and uh, Rama's presentation. So let's jump into it. The agenda is pretty simple. So what's curation at scale? Why do we do curation at scale, and how do we do curation at scale, right? So we'll basically go through each of these points in some detail. So first, basically, let's talk about what do we mean when we say what is curation at a scale, right? Yeah, so basically, so this is basically what this the product of what curation at a scale looks like. We have around 700,000 data sets on Poly, both public, premium, as well as proprietary data sets, and several different data types, and they have been curated to a similar level. And we have around 4.1 million samples uh, with around 23 data types as well as 32 data sources, and all of this is curated to a certain level and to a certain fair benchmark level on Poly. So that's what we mean when we say curation at a scale. Um, and basically how is this connected to data quality is basically as we have seen from Chef's presentation, that metadata annotation is a very important part of improving the data quality and bringing them onto Poly. It means that we are trying to add value to those data sets by improving their data quality. So in the context of this presentation, we'll look at metadata annotation in detail. There are other aspects of this as well for curation, like data reprocessing and other steps, which we'll not be discussing in this presentation, but we can discuss that later on if you want. But metadata annotation is what we consider it's a very important part of improving the data quality and bringing the data sets to the same curation level as we have right now on Omics Atlas, right? So that brings us to like, what do we mean by metadata annotation, right? So it simply is basically annotating different uh, kinds of metadata in different data sets to using same molecular identifiers, using the same vocabulary, and tagging each sample with a similar with the relevant information. So one example of that is like here we are tagging each of these cell lines with a skin fibroblast cell type tag, which is basically coming from CBCL ontology and other tags as well. Basically the uniformness of colors here indicates that we are using the same vocabulary for all of these tags. So it's as simple as that, tagging different samples, and we'll also check the other kinds of tagging as well for unstructured data as well. But this is what we mean by metadata annotation. And we'll see that how this metadata annotation enables us to unlock value of data, which is like far greater than what is currently now available on different public sources as well as proprietary sources uh, with different organizations, right? So going forward, basically, what does metadata at source look like? Is it bad, good, or what? So metadata at sources is actually pretty in bad condition. There are around like more than 50% of annotations which are missing. Only 2% of the annotations follow any kind of vocabulary. Rest of them do not follow any kind of vocabulary. So for example, in the example below, you can see that the disease is mentioned, and this was mentioned by Chef as well. The disease is mentioned, but it's mentioned in extraction protocol, and it's in a very cryptic way. It's mentioned in the source name as like TALL, which is a kind of leukemia, right? It does not follow any vocabulary, and it's very hard to figure out that what this sample is unless and until you look at it closely, individually, one by one, which is like almost impossible to do in today's world because we have so many samples in so many public sources as well as in proprietary sources, right? So that's a problem statement right now, and that really leads to a lot of uh, unusability of data that we see in today's public sources around the world, right? Um, this is another example, and also like going into more detail about like what does the problem look like. So AML might be called something else. There might be missing annotations, right? We saw this example where there's a very cryptic way of saying that this belongs to leukemia. You can see another example as well, right? Where basically the only way you can figure out that there is a disease is by looking at the source name and there is AML word written there, right? There is no other way, right? It's like a riddle. So these are some of the like big problems that metadata at different sources around the world have. And what does basically Poly offers, right? So basically, what we claim is that uh, less than 1% of annotations are missing on Poly. That means like there's a huge jump of annotations that are available on Poly, and they all follow the same vocabulary. For example, in this particular example, there's the same. There's a similar geo data set that we have taken, and here you can see like different cell types, cell lines, and diseases, as well as chemical being tagged with different ontologies. And those ontologies are followed throughout Poly across sources. That means that it need not be just geo, or it need not be just links. It will be like it will be all the sources together, and that really enables some things which we'll talk about later, right? So this is what we promise for metadata on Poly, and this is what we have been trying to do for past year. Now, what basically this means is that the scale of curation is huge, because we know that we have around 700,000 data sets on Poly, and they all have like some samples. So around what does this scale look like? There are around 4.1 million samples which are curated, around 1.2 million manually curated labels are present on Poly, around 35 million auto-curated labels, so each basically squared, and around 35 million such squares are present on in our data. And they all follow the same ontologies. That means like same colors. There are around 14 fields which are standardized across 32 different sources. Uh, that means even if it is like not geo or something else, you'll find those fields, right? You'll find disease, you'll find tissue, etc. And we are trying to expand the number of fields by 5 every quarter. That means we are growing on this vertical horizon as well. So this is the scale of curation that we are doing on Poly, and we are doing that continuously. We are doubling our data sets every quarter. So that means that this grows as at an exponential rate, right? So basically, just to summarize, there are like less than 1% annotations are missing on Poly as compared to more than like 50% annotations, and only around 2% annotations they follow the same vocabulary, while all the annotations on Poly they follow the same vocabulary or some vocabulary, right? And there are the one special thing about Poly is that Poly is a centralized location for different sources of data, even if it is like TCGA, Geo, CPTAG. If you have to like search for data, and if you have to like use data, you will have to like learn all of these different sources. While on Poly, you have to learn only one, so that basically reduces a lot of time. We'll see more benefits of those in coming slides. But this is what basically we define as like this is what is curation at a scale. Now, why do we need this curation at a scale? Right? What's the utility of this? What's the value proposition? Um, so one first line, which is a very obvious one, is searching for data sets, finding more data, right? Every time we are grasping for data in preclinical world or in the clinical world as well. But we believe that those data sets are actually available out there, and we just need to like grasp it and make it more searchable. So the problem starts with public sources, like 80% of different data sources don't have any mechanism to search samples. So that's the problem one right there, right? And out of those, like missing annotations lead to like 17% drop in results, and then there is extra 10% drop in results due to absence of ontologies. And this leads to like a very minuscule data that you get after all of this. Like you can see that there's a lot of data might be present for, uh, let's say, rapamycin, which is mentioned as sirolimus here. And in this case, you'll probably hit only one because you are using sirolimus, which is lesser known name for rapamycin, and basically the vocabulary is not standardized, right? So searching is one, and one of the things. So basically, what does it look like on Poly? So if you search for something on Poly, you not only get data from one source, but you get data from several different sources, and many times these sources are complementary in nature. For example, in Geo, you'll find diverse experiment designs, immune response, drug resistance, etc. While in links, you'll find a lot of treatment, knockout data, right? Which is very focused. And you can combine both of these together to answer a lot of questions, biological questions you have. And that basically gives Poly a leverage that you'll always get 300% more on an average, more results across sources. And you have the same key basically used across different sources. So you get same response. And that's very powerful for search because standardizing it leads to a lot more searchability and findability of data. So that's one of the advantage why we do curation at a scale. Another advantage is basically in the analysis itself. For example, let's say if you ask a question about, I want to find out what all rapamycin treatments are available in the world, be it tissue or tissue, cell line, whatever, right? You're just focused on finding all the rapamycin treatments. If you want to do it on different sources, you'll have to learn different sources, standardize them, and then find out like what is going on. But on Poly, you can basically make cohorts of rapamycin along with matched cohorts of, let's say, DMSO or any control treatment, and you should be able to do this across different sources very easily on Poly. And in our experience, basically, this is 30 times faster than the source. For example, if it will take you a month to like scout for data across different sources, while it will just take you a day on Poly, right? And imagine like there are around 10,000 such drug treatments which are available on Poly. And that basically scales it up and gives you a lot more freedom to do with a lot more data across like different kinds of drug treatments. And the same goes for like tissue, cell line, etc. So there's a lot that can be done with this kind of curation, and more than like 50 million sample comparisons which are possible. So you can do a lot of analysis at a scale. This is another advantage that one has when they have curated data. Another advantage that people have is like knowledge graph generation. And basically knowledge graph generation, not just on text mining, but also using the data points themselves, because the data is also present. Just to remind you, it's not only the metadata that is present on Poly, right? So you can see, you can possibly like ask a question like, do we have an experiment in which an AFLD is treated by rosuvastatin in liver, right? And you can see that linkage very clearly in Poly, while it will take a long time to figure that out from public sources. What is it? What data is it? And how strong is that relationship? That can be done very quickly on Poly, and you can make your own knowledge graphs from that, right? And you can create more richer knowledge graphs across 35 million auto-curated entities. So that's another advantage. So both in analysis and search, you get that advantage by just curating metadata alone, right? And imagine what you can do with data. On the same note, you can also basically use just this metadata to get structured databases. So basically curate a lot of these unstructured documents to find relationships between these entities and basically create these databases with relationships among these different concepts like tissue, cell line, disease, etc. Even concepts which are harder to curate, let's say some kind of patient adverse reaction or something is also doable, and you can create these databases, and those can then be queried by your organization. So this is basically only metadata curation. So we also do that, and we have done that for around 5 million documents so far, and we are going to do that for more. And that's basically another way to utilize whatever technology we have developed so far. So basically, sorry, what is this technology, right? And how do we do this curation at scale, right? What powers this curation? Um, so basically, the one thing that greatly powers this is basically machine learning itself and already pre-trained data which are self-supervised in nature. Now, what does this mean is that, like, and people know it by different terms. One is active learning. So you have unannotated text, you weakly annotate it using some rules or even like state of the art algorithm, and then let experts work on that. And that is much faster than only like using an unannotated text and doing it from there, right? Once you have an expert annotation, you might not have a very uniform vocabulary, but you'll have you'll have mostly like filled data with very less missing values. Once you have that, then you can use that to like train models, and which can basically standardize all the vocabulary and tag all of these different annotations together. This basically greatly powers our manual curation process, makes it faster, about 4 times faster than normal curation process. And since it is self-supervised in nature, so basically it can be automated to a greater extent. So this kind of manual curation actually powers our curation a lot. And at the core of this is basically our Polybird class of models, which are trained on around 17 billion words and have around 660 million parameters in total. And the cycle of basically generating weekly annotated data and having expert curate weekly annotated data and learn from better annotations help us to curate this data at a scale. We have done this for some fields. We believe that we can do better on those fields as well. As in order to add these new fields, we'll have to basically do better on active learning as well, right? So this is a very key piece of technology which enables us to do this at a scale. Uh, so this all about curation so far, and happy to take questions.